Hi, this is Sharon Holthouse, and I'm going to be talking to you today about nucleic acids. In class, we've been talking about different organic compounds, and there are four broad categories or classes of these organic compounds. One is carbohydrates, another is proteins, another is lipids, and this final category or class is called nucleic acids. So, primarily, I'm going to be talking to you about DNA and RNA. So, DNA stands for deoxyribonucleic acid, and DNA is found in the nucleus of cells. Each of our cells has the same DNA, with the exception of our eggs and sperm, the gametes in the body. But DNA carries the genetic code for an organism. So this code contains the instructions for how to operate the cell. And it's cells that make up our body. So these instructions are basically carrying the instructions for how to operate our bodies. RNA, or ribonucleic acid, is responsible for reading this genetic code, and then it carries the message or the instructions of DNA out to the cytoplasm where it's used for protein production. When we look at the structure of DNA and RNA, we see that it's nucleotides that are the subunits or monomers for these bigger molecules. And we talked about this concept of monomers earlier. A monomer is a repeating identical or similar subunit that's used to make a bigger molecule. So for example, with carbohydrates, we talked about how glucose monomers can be used. Several glucose molecules are bonded together to make glycogen, or a string of amino acids makes the basic structure for a protein. Okay, so the monomer or building block for nucleic acids are nucleotides. And a nucleotide has three components. It contains one 5-carbon sugar ring. It contains one phosphate group and one nitrogen-containing base. Okay, so we'll look more specifically at a nucleotide for DNA. So these are the building blocks for DNA. And we're actually looking at this diagram. There are two nucleotides. There's an adenine nucleotide and a thymine nucleotide. Let's just look at the adenine one first. You can see there's a sugar here. It's got 1, 2, 3, 4, 5 carbons. This is called deoxyribose, the sugar right here. And it is covalently bonded to a phosphate group and a nitrogen-containing base. And this nucleotide happens to have the base adenine. If we look at the nucleotide right next door, this is a thymine nucleotide. Again, it's going to have the deoxyribose sugar, a phosphate group, and this nucleotide has thymine for its nitrogen-containing base. Okay, so again, nucleotides are the building blocks, subunits, monomers. All three of those work to describe the building blocks for a larger DNA molecule. Let me just go back one second. I just wanted to make sure that I mentioned that these are covalent bonds between each of these components. So the base is covalently bonded to the sugar, or the deoxyribose, and the phosphate is covalently bonded to the deoxyribose as well. All right, so each of those subunits or nucleotides is a molecule, but when you put those units together, you get a really big molecule or a macromolecule called DNA. And DNA's structure is called a double helix. And it ends up looking like a ladder that's been twisted. So the arrangement of the nucleotides involves a backbone. And the backbone, this would be this purple, the sides of the ladder. And those are alternating deoxyribose sugar that are covalently bonded to phosphate groups. Okay, so you've got this deoxyribose sugar that is going to be bonded to a phosphate, then a sugar, then a phosphate. And so this is the way that the nucleotides are linked together. You end up getting two chains of nucleotides that make up each side of the ladder. Okay, so again, it's covalent bonds that are holding these alternating deoxyribose sugars and phosphates together. Okay, so you have these two long chains of nucleotides, and they're going to have their bases kind of hanging out or sticking into the interior portion of the molecule. And these bases form what we call complementary pairs to make the rungs of this ladder. These dots here are indicating hydrogen bonds. And so we see that adenine bonds to thymine, and guanine binds to cytosine. And we could keep going down the ladder and seeing how these two long strands are bonded together via these complementary base pairs. Okay, you have the key down here for what all these A, C, T, and G's represent. Okay, when we look at the structure of RNA, we start with the same type of nucleotide subunit. And the nucleotides for RNA are a little bit different because they have ribose sugar rather than deoxyribose. It's still a 5-carbon sugar, 1, 2, 3, 4, 5. The difference between ribose and deoxyribose is that ribose has this oxygen here, where deoxyribose simply has a hydrogen in that position. The phosphate group is still part of the nucleotide structure, and then we have a nitrogen-containing base. Now, there are 4 different types of nucleotides involved in RNA as well: adenine, guanine, cytosine, but instead of thymine that we find in DNA nucleotides, we have uracil as the 4th nucleotide. Okay, so again, these are covalently bonded together, the phosphate to the ribose sugar and the ribose sugar to the nitrogen-containing base. And we're going to see again that RNA is made of these subunits, and rather than having 2 strands or 2 chains of nucleotides, we just have a single one with RNA. And that single strand again has alternating ribose and phosphate groups that make up the backbone, or the backbone of that chain. And then these nitrogen-containing bases are kind of hanging off that backbone. And again, the sugar and phosphates are covalently bonded together. So this diagram just shows us the 4 different types of nitrogen-containing bases that are incorporated into the RNA nucleotides. The structure that I'm describing here is the structure for messenger RNA. And we'll talk later on, there's different types of RNA that are found in the body. But for right now, we're talking about messenger RNA. Okay, so RNA forms a temporary bond with DNA bases when it is reading the genetic code. Okay, so this diagram shows a bit of DNA that the double helix is opening up, and we're getting this RNA molecule that's forming as it reads the DNA. We're going to explain this process in a lot more detail, but what I wanted to highlight is that the complementary base pairs between DNA and RNA go like this. So we've got cytosine on DNA, that's going to bond with guanine on the RNA. DNA guanine is going to bind to RNA cytosine. Actually, we don't have that example shown here, but if we were to include this G right here, that would have a RNA cytosine across from it. DNA thymine binds to RNA adenine, and DNA adenine binds to uracil. Okay, so when you looked at the double helix for DNA, we saw that adenine and thymine were complementary base pairs. But in this temporary relationship, when RNA is binding onto DNA, we're going to see adenine and uracil pairing together because there is no thymine RNA nucleotide. So adenine and uracil base pair. Okay, so again, we'll explain more about this process. It's called transcription, but I did want to show you this relationship between RNA and DNA and those base pairs. So we'll just briefly contrast some characteristics of DNA and RNA. First of all, DNA is found in the nucleus, and DNA is a big molecule. So each strand of that ladder could be millions of nucleotides long. So these are very big macromolecules. RNA is most typically found in the cytoplasm. Some of it will be found in the nucleus, but it does most of its work as far as carrying genetic information for protein production. That happens in the cytoplasm. So the majority of the RNA is found in the cytoplasm, and it's much shorter than DNA. It's dozens to hundreds of nucleotides in length. Okay, so when we get into looking at how RNA reads the genetic code, we're going to see, kind of like we saw in the last slide, it's just going to read a portion of the DNA molecule, and that's why it's much shorter in length. This diagram just shows a very nice comparison of the two. You've got your DNA double helix. We've got two strands, sugar phosphate backbone, and then the base pairs in the middle coming together via hydrogen bonds. It shows all the bases that are involved in DNA. Here are the bases involved in RNA, and again, this is the single strand of RNA. Okay, so how does this relate to genes? First of all, a definition for a gene is a length of DNA that codes for a specific protein. Now, this protein may be what we call a structural protein. So we have DNA in our body that codes for the production of collagen, a very common protein that we find in our bones, our skin, our ligaments, our tendons. It's all over the body. Um, it might be a protein that codes for keratin. That's a protein that we find in our nails and our hair. Okay, so those are structural proteins. They make up um, the structures of our body. We also have a lot of functional proteins. And we talked a lot about enzymes. Enzymes are proteins that catalyze all the physiological processes in the body. Without enzymes, our bodies wouldn't function. So a gene can code for a specific enzyme. It can code for a specific protein channel or protein pump in a cell membrane. Okay, so again, there's going to be a length of DNA or a series of base pairs, and that sequence or length of DNA then goes on to code for a specific protein. Now, as we mentioned, the DNA is in the nucleus. So what RNA can do is it can read that specific gene or specific length of DNA and carry that information out to the cytoplasm for protein production. Okay, an analogy to think about is if you go to the library and you want to read a reference book. There are reference books that are very important. They're so important that the librarians will not let you check them out, but you can read them while you're in the library. And the DNA is like that. It stays in the nucleus. It's very important. It's the instructions for how to run the body. It stays in the nucleus. Okay, let's say you go to the library and you're looking at a reference book and there's a very important recipe that you want to copy out of an amazing cookbook. You can make a photocopy of that and then take it out and take it home to your kitchen and make something with it. Okay, maybe a great cake. And RNA is able to similarly copy the information from DNA, the reference book, so to speak, and then carry these photocopied instructions out to the cytoplasm, and then that information will be used to make proteins. Okay? So that's their roles in relationship to genes and protein production. All right, I want to talk about other nucleotide molecules, and we'll list 3 of them, and then I'll go into more detail about ATP specifically. So ATP is the acronym for adenosine triphosphate. And this is a direct energy source that cells use to do work. ADP is called adenosine diphosphate. And this is closely related to ATP. It has one less phosphate group. Adenosine triphosphate has 3 phosphate groups, and adenosine diphosphate has 2 phosphate groups. And then finally we have cyclic adenosine monophosphate. And this molecule is used as a messenger. Later on we'll talk about second messenger systems, but it's very important for communication between cells. So that's cyclic AMP. Let's just look at the structure of ATP. And if we look closely, we see a ribose sugar, we see adenine, a nitrogen-containing base, and it's actually very similar to an RNA adenine nucleotide, except for the fact that we don't have just one phosphate group, we have 3 phosphate groups. And again, these are covalent bonds between the nitrogen-containing base, the ribose, and these 3 phosphate groups. Now what's important is that these bonds between the phosphate groups are high energy bonds. you can see that these phosphate groups have a negative charge, and we know that like charges tend to repel each other, okay? But these bonds, you can think of trying to shove together two things that um, might not want to be together. It's almost like um, shortening a spring. Okay, so there's a lot of energy that's captured in these phosphate bonds, and that spring is kind of is ready to um, lengthen and release energy when cells use ATP. Okay, so ATP is like tiny packets of energy that are readily available to cells. And if we look at this ATP-ADP cycle, what happens is we have an ATP molecule, and with the addition of water, we end up with ADP, adenosine diphosphate. So in other words, we're just taking off, we're breaking this bond right here, okay? When that bond is broken, we get the release of energy as well as a free phosphate group, okay? So when the body's using a lot of ATP, when it requires energy for a muscle contraction or to run a sodium-potassium pump, all sorts of physiological processes in the body, then we're going to end up with more and more ADP. Well, what the body can do through a series of reactions, this phosphate group is actually added onto this ADP, and we're able to produce more ATP. This process of forming ATP is called cellular respiration, and we'll talk a lot more about that later on. But basically, our body uses energy from nutrients such as glucose, and through a complicated process, many steps, many different enzymes, it's able to use that energy from glucose, for example, to form this bond between this phosphate and ADP to get ATP. Okay, so our body's constantly breaking down ATP when it needs to do work, and it's constantly forming ATP so that it has it readily available for energy for the physiological processes. So you might be wondering, why is ATP such a great energy source? We obviously eat food for energy. We eat carbohydrates and proteins and lipids. But if we just think about ATP as an energy source compared to, for example, glucose, ATP is really ideal. It's readily available. It's convenient because it just requires the breaking of this one bond between the last two phosphate groups. As I mentioned, those are high energy bonds, so you get a significant amount of energy that's released. And this process only requires one enzyme. If we were to use glucose for energy, it would take several enzymes, and cells need energies quickly and all the time. So having these little ATP packets available is a much more efficient way to get energy. Finally, ATP is found throughout the cell. Glucose is the enzymes that are needed to break down glucose are generally only found in the mitochondria. But this one enzyme that's required to break the phosphate bonds is found throughout the cell. So again, it's a very available, convenient energy source for cells to use for all sorts of processes in the body. All right, so that concludes our discussion of nucleic acids and nucleotide molecules. And I hope to see you soon and hope you have a great day. Thanks.